It's been a great day. I've really enjoyed it. It's fascinating talk. It's a challenge being the last speaker. There's an old joke about being Elizabeth Taylor's 8th husband. Do you know this joke? You know, you may know what you're doing, but it's pretty hard to do something original. But I'm going to tell you a little about start off with some of my basic research, which is Interleukin 7, which is what got me into this business. So I'm a basic scientist, not in the clinic. This is what Interleukin 7 does. It's required for T-cell development. This is a thymus of a mouse, and this is a mouse that doesn't have the receptor for Interleukin 7. We have patients like this. So this is severe combined immunodeficiency, is a deficiency in this pathway. Right? So it's absolutely essential for T-cell development and survival. So this is, again, Interleukin 7. This is the receptor, 2 chains. This is a kinase called Janus 1 and Janus 3. This is the alpha chain, the gamma C chain of the receptor. And that's what IL-7 normally does, is it brings these two together. These are 2 enzymes. They phosphorylate one another, and they start the signaling cascade. So my lab spent a long time doing basic science with this IL-7 pathway. And what it keeps cells alive, and it does it by protecting mitochondria. That's mitochondrion. These are death proteins coming in that lyse the mitochondrion and trigger the apoptotic cascade. They're protective molecules, the mitochondria, and they're death proteins that are in red. So this is the death pathway, and IL-7 critically keeps T cells alive based on balancing these factors in mitochondria. So it protects from cell death. We later learned that it is a mitotic factor if there's enough of it around. And the way it does that, I just zip through it, but the way it does that is through controlling ultimately a CDK, which controls cell division. So it controls cell death, it keeps them alive, and it can make them divide. And so I've worked in the Cancer Institute for many years, and finally something came along that connected my research to cancer, which was that it turns out it causes leukemia. And you've heard a lot about ALL. This is what we had found about 5 years ago now, that was that there are mutations in the alpha chain, IL-7R alpha, in about 10%. These are 3 different European cohorts. And the rest of the ALLs, these are TALLs, most of them probably either use IL-7 itself or have mutations in other parts of the pathway. But the actual activating mutations are in about 10%. We're not the only ones to find this. There are groups at St. Jude's and groups in Israel and in Europe as well. So activating mutations in IL-7R alpha are drivers, and the pathway is probably very common in this. You already heard definitions of ALL. It's the most common childhood malignancy, 3,250 estimated cases per year in the United States. There are 2 major forms, B-cell ALL being the more prominent. TALL is less common. That's the one I'm mainly going to talk about. But IL-7 pathways also looks to be very important in B-ALL. Initially, there were the earliest connections were with Notch. Notch is an interesting membrane protein which creates a transcription factor when it's cleaved. And that was the first gene associated with it. Later on, the PI3 kinase pathway was associated with it. And so now we're implicating the IL-7 pathway. These are the mutations in the receptor. No 2 patients are identical. We have hundreds of them now. And they're very interesting mutations. They are insertions. And the insertions, which are very uncommon, you've heard about translocations and point mutations and so on. These are insertions, in-frame insertions, and they occur here at the border of the extracellular domain and the transmembrane domain, right at the border. These are 2 typical patients that we worked on quite a bit, patient 1 and patient 7. And what almost all of them have in common are cysteines. So here's a cysteine here in the insertion in patient 1, cysteine here in patient 2. Many of them have prolines. And in the ones that we've really more thoroughly investigated, they need both of those in the insertions, both the cysteine and the proline. What they do is create constitutive signals. And so this is looking at the wild-type receptor with IL-7. It induces a signal, which is phosphorylation of a substrate downstream. Take away IL-7 and it goes away. But if you put in mutant receptors, patient 1 or patient 7, it keeps signaling. It doesn't need IL-7 anymore. So it's ligand-independent signaling. That's what the mutation causes. And yet another surprise was that it didn't even need the other chain. So normally IL-7 signaling needs the gamma-C chain, and it needs another Janus kinase. It doesn't need it in the case of these ALLs. Right? This is what that mutation does. So I said that there are cysteines. What cysteines can do is form disulfide bonds, and these are homodimers that are created by those disulfide bonds. In a Western blot, now a reducing agent is added, and they're reduced down to the monomer size. So that's what the cysteine does. It also creates a leukemia. It's sufficient to create a leukemia from an immortalized mouse cell line. So kind of like they used to use 3T3 cells, put in an oncogene, and you would get a tumor. That's kind of what we started with here. This is the mouse cell line. We put in patient 1 or patient 7 IL-7 receptors, and we saw leukemias in mice. And this is like the money shot right here. This is an oncogene expressed on the cell surface. It's on the cell surface in both the primary and in the distant metastases. That meant that we could use an antibody to kill the tumor cells. So you'll see that develop later on, and that was what CCF funded for us. Well, this was the primary tumor under the skin in the mouse that we created with this oncogene, and then it goes all over. It goes to all the places that ALL goes in a child. And the signaling, again, is what intrigued us as basic scientists for a long time. So that's how IL-7 normally works. It brings together the two chains, the two enzymes come together and start phosphorylating everything. What the cysteine does is it makes the dimer, but the dimer is not enough. You have to have something else. And so we think what the proline does is reorient the inside of this receptor so that it can bring the two kinases together. That's purely a model, but we're trying to model it actually with more sophisticated methods than using sticks like I did here. And now, this is a little more basic science, the kind of thing that my lab does mostly. So IL-7, mutant IL-7 receptor is an oncogene by the classical criteria that it transforms a cell line. This is a mouse thymocyte line, and it generates leukemia. But like almost all oncogenes, or most oncogenes, the receptor is really insufficient to transform a primary cell. So if we take a T cell or a T cell progenitor, we put it in this mutant IL-7 receptor, we don't get a leukemia. We get some weird myelodysplasia, but we don't get a leukemia. So we've been looking to see what actually would work together with IL-7 receptor. And we noted that in one extensive study that had been done, that in about 15 patients with ALLs that had this IL-7 receptor mutation, about half of them had mutations in the RAS pathway. So we've tried to complement it with various things, but I'll show you the one that actually works. We put it together with RAS, and indeed, that's what we see. So these are mice, spleen. This is the spleen of a mouse, greatly enlarged, if you've never seen one before. And these are even more greatly enlarged. Yeah, they're gigantic. That's splenomegaly by your standards, I guess. So that's what we got. So we got cooperation between mutant IL-7 receptor and RAS by the criteria that it made these big organs, but more important that it created leukemia. And so these are the final blood leukemia of the first two mice. But we've done 20 or so of these now, and they all generate leukemias. So RAS is sufficient, together with IL-7 receptor, to get a T cell acute lymphoblastic leukemia. And that means, the significant being, I don't know if I put that on a slide, the significance would be that we could target the IL-7 receptor pathway, as I will show you, and may be able to target the RAS pathway. As you may know, NCI has a very large program to try to actually do just that, to target the RAS pathway. And we'll get together. So this is the cartoon that we just recently did, but this shows various ways that you could attack this whole signaling scheme. So there were various mutations all through here that I'm not showing, including the receptor that we first found. but others have found various other components of this, which also will give a leukemia. But I'll show you the two ones that we're working on, which are antibodies to this chain itself, and enzyme inhibitors, Janus kinase inhibitors. So there they are again, Janus kinase inhibitors and anti-IL-7 receptor antibodies. Why would we do this? We've heard a lot about why you would want to do this today, but the cure rate of ALL is one of the great success stories in modern medicine. It's 80% today, but as you've seen before, the toxicity is very great. I'd love to have that long list of all the terrible things that happen to patients who are treated with chemotherapy. But this picture really got to me. A colleague sent this. These are identical twins treated in England, and it kind of illustrates the toxicity of conventional chemotherapy for ALL. Well, these were two kinase inhibitors that we could work with. This one from Pfizer, and this one from Incyte. Also, Novartis has bought this. And these are FDA approved for other indications. They both work in vitro. And here we actually start getting funding from the Children's Cancer Foundation, which is why I put this in on this slide, because this, so the folks that make these are not, in fact, I was thinking this morning about the talk, they're not much interested in childhood cancer. They're interested in treating old people for a long time and selling a lot of drug. And so they actually would not give us the drugs. And we had to buy them. And now actually we're synthesizing it, this one at NCI. And so we had to buy them, and they were very expensive. They work great in vitro. This is just titrating against cells in vitro. They're more toxic than you would gather from reading any of the literature. And so we had to titrate in both of these drugs to the toxicity point. This one turned out to be the promising one. So there was a therapeutic window with ruxolitinib. It's a better JAK1 inhibitor. This was what we did with it in terms of the treatments, 5 treatments here after first putting in the artificial leukemia. And it showed some efficacy as a single agent. This is lung, blood, and spleen. This is the artificial leukemia. This is where we have a mouse cell line that's transformed really by putting in the mutant receptor. So we then turned actually to a real human xenograft, and we got it actually locally. So Kurt Sivan at University of Maryland had this growing in these extreme immunodeficient mice called NSG. This TAL number 5 expresses the receptor, it responds to IL-7, and ruxolitinib blocks the response. I think I have that in here somewhere. So this is the expression of this receptor on this TAL. And this is the response to IL-7, shown here. This is phosphorylation of a downstream substrate. And this is a very nice effect of the inhibitor in blocking that signaling response. But it didn't work. And so probably the window here of toxicity is just not favorable. So we treated, I'm losing it, a number of times and really could not achieve any significant effect as a single agent. The promise, though, is that maybe putting it together with a chemotherapeutic agent, which is actually in use in ALL, will work. So it works in vitro, and these are experiments that we're doing now. So you put the two drugs together, and you do seem to see some promising effect. So ruxolitinib may be insufficient as a single agent in TLL, but maybe combined with chemo or other biologicals will have better success. But now I'll turn to the one that really does look promising, and these are antibodies generated originally against the mutants. So our collaborator Scott Walsh at the University of Maryland is a crystallographer who's great at purifying proteins, and he provided these the antigens that we then generated monoclonal antibodies against, and so that turned out to be a much more formidable problem to generate these antibodies than I thought it ever would be. But it was. We're not Pfizer, you know, and so we have a research lab. We did finally come up with 2 antibodies that are called here, 2B8 and 4A10, and a little bit of things about them. They react both with the mutant and the wild type. We didn't really think we would get a mutant-specific one. And if we did, every patient is different, so I didn't really think this would necessarily be an advantage. We didn't get it anyway, but they work against both. They work against normal human T cells. They work against monkey T cells, fortunately, so we can do toxicity studies. They work on most TALLs in the sense of binding to them. They don't compete with the ligand. They're not rapidly internalized. And fortunately, the two recognize different epitopes. And one way, yeah? The second. So they are two independent proteins that are physically associated. Okay, right. But it's a very selective association between the two. And it seems to be the only kinase that's really important in the intracellular signaling. So we then converted them also with help from, so we made them with help from this foundation, and we converted them with help from the foundation to what's called a chimeric antibody. So this is like ruxolitinib, which is used and been mentioned several times against lymphoma. And the reason for doing this, so this is a starting mouse, all in blue. We converted them to sort of 3/4 human, kept the V regions. And the reason for doing this is not just because you want to put them into patients, but because this end of the molecule is very important in therapy. And it turns out the mouse antibody doesn't work well even in mice because it doesn't bind well to the FC receptor and trigger antibody-dependent cell-mediated cytotoxicity. So the experiments that I'll show you are actually with this chimeric antibody. This is the BIA-core. I think we heard about the BIA-core. This just shows that they bind to different things. The two antibodies bind to separate epitopes, which may prove useful in the future. And this is another way to look at it. So the first antibody gives this much fluorescence intensity. The second one gives that. And combined, they give that. So they're binding to different spots, different epitopes on the IL-7 receptor. Okay. And so we're using them in for ADCC, antibody-dependent cell-mediated cytotoxicity. We've seen cartoons of this before, but the idea is that they bind to, in this case, IL-7 receptor. These are the antibodies binding. This is usually an NK cell. They're the best killers, using their FC receptors to bind, and then releasing various toxic molecules and killing the target cell. So the chimeric antibodies work quite well in vitro. So this is using them against first one of our artificial lines, which is a mouse line with actually in this case, a wild type IL-7 receptor, titrate in human NK cells, and you get lysis of this target cell. This is against the mutant IL-7 receptor, showing that it also serves as a good target to human NK cells. And this is an actual TALL. This is a human cell line, which has a mutant receptor, as a matter of fact. It's one shown here. Now, the cell line is actually killed to some extent by NK cells. So NK cells have the capacity to recognize some leukemia cells without any additional antibody in between. But the antibody facilitates it, so it gets better. So they can recognize the human ALL. Now, the downside is, and this is what I probably should have said 10 minutes ago, is that the expectation is that they would also kill normal human T cells. Because most human T cells, when they're naive, at least, or when they're memory, have IL-7 receptor. And sure enough, we're titrating in human NK cells against human T cells together with the antibody, and it does kill them. So the expectation is that there will be a lot of T cell killing. Before we ever started this, we anticipated that would happen. And so we met with the pediatric oncology folks, and they said, that happens anyway. The way we do it, usually we have this problem. These are children. They have the ability to regenerate their T cells because they have a thymus. So this is, they tell me, an acceptable toxicity. So here's the therapeutic effect in various types of mouse models. So in this case, it's the mouse cell line transfected with a mutant receptor, put in here. One injection of the chimeric antibody here, and then look at various responses downstream. And the response was really quite good. So this is looking at the spleen of such a mouse. So these are 3 mice with a lot of these artificial leukemia cells, and this is effectively clearing them from the spleen or the liver. So this is the positives, and these are responding individuals. Okay, and effect on organ sizes, so that these enlarged organs, also affected by antibody treatments. And this is effect on survival with 1 injection of the antibody here. This is the controls dying here, or they're sacrificed. We don't allow them to die. And these are various concentrations of antibody extending survival. So, keep jumping. So let's go back to the real human ALL from Kurt Siven. I showed you this before. It expresses IL-7 receptor. It responds to IL-7. And it grows in mice. And this is an experiment that shouldn't have worked at all. And I'll show you that it does work with caveats. It shouldn't have worked because we thought the only way to grow these was in what's called the NSG mouse. Well, the G stands for gamma common. And what gamma common receptor is part of the interleukin 15 receptor. The result being that there aren't any NK cells in the mouse. So we didn't think this should really work at all or very well, or at least in retrospect we should have thought that, but we didn't necessarily think so thoroughly at the time. But it does work pretty well in that this is the control. This is treating such a mouse, putting this human ALL in, even with no NK cells. And this is the chimeric antibody working pretty well in 3 of these individuals, looking at the spleen or looking in the blood. So this is a real leukemia, compared to the controls. So it does actually work pretty well, even without NK cells. And even in the liver, very similar. But what we didn't succeed in doing much was in the bone marrow. And so we heard a little bit about the magic of bone marrow. This doesn't really show it very well, but there's probably about a 25% residual leukemia cells in the bone marrow of these mice. This isn't a very quantitative looking thing, but if you can read the numbers, it looks something like that. So it clears it pretty well in the other organs, but not very well at all. And there was no real survival benefit of it. So we happened to see this, and it came up when we were talking about your study, was that these cells express a lot of CXCR4. And that's a lot. That is really a lot of this chemokine receptor. And so my technician, Julie Hickson, really thought of this. I give her all the credit. She said she had read something about leukemia cells hiding in the bone marrow and being protected. And she found this, and CXCR4 is the receptor for the chemokine SDF1. It retains cells in bone marrow, and it could impart survival signals to TLL. It apparently does this in CLL. So Julie's idea was to put in this antagonist, AMD 3100, which competes with SDF1. It's an approved drug for stem cell mobilization. And it worked great. It mobilized these ALL cells out of the blood, out of the bone marrow, into the blood. They're coming out here, and they're getting killed, we're looking in the blood now, by the antibody. And the result was also very nice in the bone marrow. So this is the antibody alone being relatively poor, and this is combining the antibody with the chemokine antagonist. So there is hope even with no NK cells around. But that's probably not where we're going to go. It's nice to know. We don't really know how this works. We don't really know how it works. And it's relatively protective. So their treatment was back here. We're extending life by this combined treatment out, kind of out to here in these mice given a real human xenograft. Right? So this was called the Invention of the Year at the University of Maryland by my collaborator, Scott Walsh, got a good review. He's up for tenure, so this is going to be good for him. And anti-IL-7 receptor is effective against the xenograft, synergizes with the chemokine antagonist. So in the future, we need to add in K cells to this, and I think that should make it work a whole lot better, even though it seems to work pretty well now. We're going to explore how the killing works. We really need to expand the panel of xenografts, and we have a collaborator in Australia who specializes in this kind of thing that will be doing it. We still haven't gotten to our second antibody, which may be some advantage. We're applying for clinical development through the NEXT program, which NCI has, run by these folks, and NIH has made a patent application, and we'll be looking for licensing of it in the future. Okay, overall, the IL-7 receptor pathway seems to drive many of the pediatric TMB ALLs. The Janus kinase inhibitor is partially effective as a signal agent, but the antibody looks like it's highly effective in the xenograft. This is the pathway again. There are mutations all through this. This was the IL-7 receptor here. Terry's left, but he works on this other one, which is called TSLP. That is this one over here. And that also shares the IL-7 receptor and could be a target for some of these treatments. Okay. Some folks from my lab, Wenxing Li really runs the lab. She's a staff scientist. Julie Hickson is way smarter than me and came up with this chemokine antagonist idea. And a lot of other folks. We've had a lot of collaborators. My friend Scott Walsh at the University of Maryland, Joao Barata and I started this. He's at Lisbon, in the University of Lisbon in Portugal, and Andre Yunus has sent us a number of really smart students. Priscilla did the original genetics, and Gisele is with us now doing some biology experiments. And thanks to the Children's Cancer Fund, one of several sources that we have income. I'm an employee of the National Cancer Institute, so we get our main funding source from them. But without seed money from Children's Cancer Foundation, we really couldn't have generated the antibodies that I think look quite promising in the future. So I made it.
So I have a question. Since I'm holding the microphone, I get to ask the first question. Is that insertion, is it a genomic insertion, or is this potentially an alternative splice site?
It's not an alternative splice site. It's in the middle of an exon. If it did splice there, it would have brought in something that we could recognize, and there is no such sequence that we can recognize. So, but it's very unusual, as you probably know as a cancer guy, but to have these in-frame insertions. And they're all like that. There are hundreds of them that are like that now. So the only real speculation that we have that is sort of clever is that there's an enzyme called TDT, which is a marker for these cells. That's terminal, if you're old enough, terminal deoxyribosyl transferase used to be used to add like bases, like radioactive bases onto nucleic acids. These cells, these immature lymphocytes have that, and that's exactly what they do. What the enzyme does is it inserts bases during BDJ recombination, and that's exactly the stage of these immature cells, both T and B. So we've staggered a bit with that idea, and don't have any real data from it yet.
Very interesting. Yeah. Gotcha.
So as you know, a lot of these different cytokine receptors, chemokine receptors have different components to them. For instance, IL-2 has the high affinity IL-2 receptor. And so interleukin-2 has been used at ultra-low doses to stimulate Tregs for graft-versus-host disease, which you would think by giving IL-2 would make GVHD worse because it would activate the T cells. Mm-hmm. I'm wondering if you can do the same thing with your antibody. Is the IL-7 expression higher in the tumor cells versus a normal T cell? And if so, could you titer your antibody to just affect the tumor cells and not the T cells?
In some cases, yes. In other cases, no. So what we see is a wide spectrum. We've got one cell that makes an astonishing amount of IL-7 receptor and TLL cell, and we have others that are down in the range of normal. So certainly, if we could find a patient, who needed treatment, there may be some out there. They almost all have it, and they almost all respond. But the range is, there can be vanishingly small amounts, and they'll still respond to IL-7 itself. There are other mutations which probably affect the internalization, We just found one the other day, actually, which looks like it affects the clathrin-mediated endocytosis of IL-7 receptor. And when that's mutated, it looks like there's even more. So it's possible that some of these that have extravagantly high levels of expression have an additional mutation in the internalization pathway.
You just say that normal IL-7 receptor actually gets internalized.
Yeah.
Then why doesn't your antibodies get internalized? Is it just the properties of those antibodies, or what?
Well, they don't get so the normal internalization to a clathrin coated pit would require ligating these both receptors and signaling, and that affects the internalization process. So the antibody per se is probably it slowly disappears. So if you look the next day, then there's much less of it bound. So if you bind it on Monday and you look on Tuesday, then there isn't as much as there was on Monday. But whether that degraded and fell off or whether it was internalized, I don't really know. But it doesn't trigger rapid internalization like the natural ligand actually does itself.